This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, Communications Director at Feinberg. Since SARS-CoV-2 was discovered in Illinois over a year ago, Feinberg scientists have been tracking the evolution of the disease in the Chicago area. Ramón Lorenzo-Redondo, Research Assistant Professor of Medicine in the Division of Infectious Diseases, is part of the team leading this work. He joins me today to talk about this research and the new COVID-19 variants and how the vaccines on the market today stand up to them. Thank you so much for joining me today.
Thank you so much for having me. It's a pleasure.
So you are originally from Spain. Tell me about your journey to Northwestern and to this remarkable team studying COVID-19 in the Division of Infectious Diseases.
It's been a journey. So I did my PhD in Spain, in Madrid, where I'm originally from. And then I came as a postdoc. I decided to join Northwestern because, I studied and I still study HIV virus. So one of the best centers for HIV was Northwestern, and I wanted to come to the US because it's probably one of the best places to do research. So I came to Northwestern and I joined as a postdoctoral researcher in the laboratory of Dr. Steven Bolinsky, and then I became after a few years I became a research assistant professor in the ID division, and then I've been having a lot of support from the CFAR, first all HIV, so from the CFAR led by Dr. Rich Aquila, and then a lot of support from the ID division led by Dr. Taibo, and also collaborating a lot lately and several grants with Dr. Thomas Hope in the Department of Developmental Biology.
Share some background with me on how viruses mutate and evolve.
Yeah, so we can think about viruses as a copying machine. Their objective in life, let's say, is to copy themselves, to produce copies. But the thing is, these viruses follow a very different strategy than more complex organisms. They have a very small genome, as opposed to our genomes that is very big and very, very complex. And what they do is they replicate a lot. They try to replicate a lot as much as possible. But when they do this, as opposed to us, they generate a lot of errors. So sometimes I like to think about this as a book, a very complex book. We are a very complex book. So to retain all our information, we need a very good copying machinery. But on the other side, the viruses, what they do is imagine these short sentences. Sometimes the vowels are changed by numbers and we still understand what they say. This is basically what the viruses are doing. They are trying to replicate as fast as possible and generate a lot of diversity. And how they do that is they copy, but they copy with a machinery that is not that good. So they make mistakes. Most of these mistakes are bad for them, but some of them stick. And they help them generate a lot of diversity, a lot of mutations. And when they generate these many mutations, they are ready to adapt really fast. So if there's a change in the environment, they can adjust just because they have the possibility to acquire a mutation that helps them adapt, or even this mutation already exists. They generate like a cloud of mutants, and they are able to adapt very fast. So they are copying, they don't care if they make mistakes, but that's how they evolve. They evolve very fast thanks to these mistakes that they make, but they help them to adapt.
Well, how does the mutation rate of SARS-CoV-2 seem to compare to other viruses you've studied, like HIV or influenza?
This virus, that is an RNA virus, as the other that you mentioned, these RNA viruses mutate a lot. They are the fastest in mutate and the fastest evolvers in nature that we know of. But this virus, as opposed to the others, has a protein that kind of helps to improve how the replication machinery works, or let's put it in this way, makes the virus make less mistakes. So that makes the virus evolve a little bit slower than other viruses. So comparing, for example, with HIV or flu, that are two of the highest mutation rates that we can find, this virus has a mutation rate that is about between 4 and 6 times less than the HIV and flu viruses. This is good news in theory, because while HIV or flu mutate so fast that forces us, or HIV, we haven't even found a vaccine. And for flu, it changes so much that every year we have to remake the vaccine. For this virus, this might not be needed, but also it's puzzling that it has this what is called proofreading mechanism in which the mutation rate is a little bit slower. And this is puzzling because in an evolutionary perspective, this has to have a reason, a biological reason. Everything that happens in biology has an advantage for the organism. So why this virus is mutating less, it's a little bit puzzling, but it might be due to faster replication. So what this virus wants is to replicate as fast as possible and then infect as many people as possible. And it doesn't need to adapt that fast because it's not encountering the immune system as much as the other viruses. But this is still questions that we need to answer. But in summary, and to be very short, the mutation rate of the coronavirus is between 4 and 6 times lower than these highly mutating viruses like flu or HIV.
And we're still learning so much about SARS-CoV-2, but I want you to take me back to last year. When did you first receive samples from people infected with SARS-CoV-2, and what did you discover right away, right off the bat, about the origins of the virus that first appeared in the Chicago area?
So we formed a small team, and we started to collect samples. So right from the beginning, we started sequencing these samples. Our first sequencing run was, I think, late March. And this is when we started sequencing the virus. So we were seeing numbers growing in a very, very high pace. And what we started to do is we need to understand the forces behind the evolution of the virus. And for that, we need to sequence the virus. So we started sequencing in March, and we had a fairly complete data set by the end of May, where we could now understand what was the origin of the epidemic and the first changes in the epidemic in the Chicago area. And what we found is that right from the beginning, we had a very diverse epidemic in Chicago. We had viruses that could be linked to viruses that came directly from China, and viruses that came through Europe and then East Coast to arrive to Chicago. And this is very interesting. It makes Chicago a very interesting place to study the virus because it's one of the mostly connected airports in the world. So because the air travel has been so important to the spread of this pandemic, we thought it was very, very important to study the virus in Chicago. And what we found right at the beginning is that because the numbers were so high, the virus was able to evolve and adapt better and generate better mutants. And these were the first variants already interesting in these studies right at the beginning, where we found that the variant or the lineage that was coming from Europe through the East Coast had acquired some mutations that made that virus probably infect or spread better. It didn't make the infection more severe, but it made the virus to spread better. And this we were able to do or to check by comparing the viral loads that the patients showed. And what we found is that the new variant that was coming through Europe and that was becoming dominant and became overwhelmingly dominant had some mutations that made the virus generate more viral load and probably spread better due to this fact.
Describe to me the process. You talked about how you analyze these specimens from the positive COVID-19 tests. Just kind of walk me through that. What equipment are you using? What does that look like? How long does it take to analyze these specimens?
One that we've been more generally using and that where we can perform these what we call molecular surveillance studies. So we understand how the virus is evolving in the population. So what we get is samples from the hospital that go to the diagnostic lab in the DMB lab in the pathology and microbiology department, and then we get when they get tested there, they get the positive samples. When they get a positive sample, we go and we get a part of that sample. Our technicians that are doing a great job, especially Lacey Simmons, now starts processing these samples in Dr. Holquist's lab. So what they do is they extract the RNA from the virus, and then they copy this RNA into DNA, and this is for technical reasons. These DNA are amplified, and then when this DNA is amplified, it goes to Dr. Oser's lab, where it gets sequenced. At the beginning, we were using a technology that is called nanopore that can sequence big portions of actually the full genome of the virus, but we could only do 24 at a time. Then we switched to another technology, and now we are using both. A technology called MySeq, so it does deep sequencing. So we get millions of reads from these viruses. So now we can also assemble full genomes, but also we can get the what I was telling you, the mutant cloud. Every virus generates a mutant cloud. They are not all the viruses that we get, even from one patient, are exactly the same. They always have some slight mutations. And so now we can understand not only the full genome and the whole genetic material of the virus, but we can also understand the intra-host, the intra-patient different variants. Then those sequences get analyzed by me, what is called phylogenetic studies. Basically, we build family trees of the viruses, and we understand how they are evolving. We see the evolutionary forces behind the evolution. And also we can do models relating to geographical, temporal data, clinical data. So we can build complex models to understand the full evolution of the viruses.
How important is the relationship between Feinberg and Northwestern Memorial Healthcare in this research? You're getting these samples from the healthcare system. Talk to me about that.
Well, that's been key. That was the only way we could understand the pandemic in Chicago and related to the global pandemic. It's very important that through Northwestern Memorial Health System, we get the samples from the patients. There can be many types of patients where we get those samples that get tested in the DMB, so that's really important. The microbiology lab also gets some samples and also helps us with this analysis. And then we get several sources, so we can get a broad range of viruses, and we can understand different questions of the virus. So not only we can understand how the virus is evolving in the population, but also again, through the Northwestern Memorial Healthcare System, Dr. Ison is building a longitudinal repository. So in this case, a longitudinal repository means from the same patient, we get several samples. This is very important, not only to understand how the virus is evolving generally, but how the virus evolves inside a person. So we can understand processes of evolution within a person. So, for example, how the virus evades the immune system, or how the virus responds to some treatments, or how the virus generally adapts to a person. So all of these questions couldn't be answered if we were not closely collaborating between the clinical part and the research, even basic research. We are doing a lot of things with understanding of basic models, evolutionary models. So if we put together clinical side with kind of abstract models, we can build very, very powerful tools where we can now study the virus and maybe predict how the virus is going to evolve, and then try to get targets or ways to fight the virus.
It's fascinating. And you mentioned now there's these new variants that keep showing up. I mean, just last month, the Chicago and Illinois Departments of Public Health announced that your team discovered another fast spreading variant, which appears to be the same one that's overtaken the pandemic in the UK. Tell me about that. And what other novel variants have you seen or you were able to report on?
As we talked before, these viruses continuously evolve. They continuously change. So what we are seeing is that when some of these mutations translate in an advantage to the virus, in a moment where there's sadly there's a big prevalence, so all these viruses are competing with each other, and at the end, what it makes the virus do is to become better. By competition, by generating mutations, they become better. So we are always going to see these viruses evolving and changing. They continuously change. That's their nature. However, what is happening is that sometimes a mutation might be so advantageous that generates what is called a bottleneck in the population. So this mutation makes the viruses so much better that it generates a new family, and this family spreads more than the others. So this is how these variants appear, where some of the viruses share some mutations. But even inside these variants, there's changes between the viruses. It's very unlikely that two viruses are exactly the same. So they always change, but they share mutations. So what is happening now, and what we are seeing, is in some places where they have very big prevalence, some of these variants are becoming dominant. And that's an indication that they might be better at replicating or better at something that makes these variants kind of dominate the population. A specific variant, the UK variant, was first reported in the UK, and what was observed is that it became dominant super fast, and it had a lot of mutations, more than expected for this virus. Suddenly a lineage appears with many mutations without a continuation of time that we could see between the previous lineages and this lineage. So that was already suspicious, but then it became very dominant, and then it started to spread all over. It's still not the majority of the sequences in around the world. It's only spreading very fast, but it still is not dominant. So again, thanks to the hard work of this small team, and in this case, regarding this new variant, we were pushing to try to find it here in Chicago. We were working nonstop, especially Lacey Simmons, that is a technician in Dr. Holquist's lab, was working nonstop to try to get this variant. So I think we have to give a lot of credit to Lacey. So Lacey was processing samples, Dr. Holquist and Dr. Oser doing all the work to sequence them, and then me analyzing them. And what we found is later in December, we found finally one of these variants. And we were expecting to find it because it was spreading so much, and the UK is highly connected. But still in the Chicago area and in general in the US, it's not dominant. But it's starting to be concerning that some of these variants are sharing some mutations, meaning that these mutations might be important for the virus and might make the virus better. And the pace they are increasing in the population is also a little bit concerning. It's not a very scary thing. And again, the same prevention measures work for each of these variants. If you wear your mask, if you do social distancing, this is not like the movies. The virus mutates and now it does something different. No, they just transmit much better. So we have to be more careful. And what this is showing us too is, first, these variants in an interconnected world go very fast, travel very fast. And secondly, because of this high prevalence, we are allowing the virus to become better all the time. So we need to lower these numbers, because with big numbers, the virus is going to find new mutations that are going to improve its capacity of infecting and maybe at some point to evading immunity and other things. So all of this evolution, more than the specific variants that we have to keep track, what it's telling us is that we are allowing the virus to become better. And this is how the viruses become better, by replicating, generating diversity, and selecting mutations. So the main message is that we need to lower the viruses, the prevalence, because if not, the virus is going to keep improving. They keep evolving all the time.
Are the current vaccines for COVID-19 effective against all of these new variants that keep showing up?
There's some studies that suggest that the vaccines, the immunity generated by the vaccines might not be as good as with other viruses with some of these variants, specifically variants that share one mutation, also in the spike protein. Some of the now famous variants that have this mutation are the South African and also one of the Brazil variants. So these are variants that they are not from the same family, but they have a same mutation. And this is what it's called, there might be a case of convergent evolution, meaning when you see the same mutation in two different families, in two different lineages of the virus, that might mean that that mutation makes the virus better. An example of this, very maybe a silly example, is the animals in the Arctic, where animals that live in the snow become, they develop white fur. They don't come from the same families, but they develop a similar trait because that helps them in that environment. So this mutation would be a little bit like that. It looks like this mutation allows the virus to be less recognized by the immune system. Still, they are recognized, but not as efficiently as other variants. And we need to understand this. I don't think we should totally alarm, but we need to keep track of these variants because they might make the vaccines less effective, not completely ineffective, but maybe less effective. We still need to understand this. But the good thing about these vaccines, especially the RNA ones, is that they are new system of vaccines that can be changed in a fairly easy manner. So now, for example, Moderna, it's already updating or changing a little bit their vaccine to cover these new variants. As long as we keep track of them, and again, if possible, if we lower the numbers, then we won't allow the virus to kind of adapt as easier as it's doing right now. So still the vaccines are going to work so far. I think they are our best hope, and they are working great, and we need to push for that. But also, it's very important to keep studying the viruses. Once the vaccine is more generally used and more population has it, that will become a pressure for the virus to evolve. So the virus, by encountering people with immunity, it will try to escape that. And we need to keep track of that. If the vaccination is spread and it's going well, we won't give the chance to the virus to evolve. But if not, the virus will try to escape this immunity and maybe try to evolve a little bit more to become less affected by the immunity. So that's why it's very important that we keep studying the virus even after vaccination to see if the virus is changing in a way that might be concerning.
Well, no doubt you will be busy here in the coming months monitoring these variants that are circulating. Are you going to have time to work on any of your HIV research?
We have to keep, HIV is still a huge problem. We tend to forget it, but no, HIV is there, and HIV is affecting and millions of people. So yeah, we keep doing HIV and other viruses. there's not that much time to rest, but also we need to keep doing some of the HIV studies. So we are doing some studies, and I'm also part of some studies with Dr. Hope, in which we are also trying to understand complex interaction between immunity and intra-host evolution, both COVID and HIV. These viruses in some ways behave very differently, but in some ways behave similarly. So we need to understand these evolutionary processes, because at the end is how the viruses adapt. And if we don't understand that, we cannot fight them, because we won't know why the viruses are changing and how the viruses are going to change.
Well, thank you so much. You gave us so much clarity and insight today, and I know that you are busy, and I appreciate your time.
Thank you so much. This was great and a pleasure. Thank you very much.
You can head to feinberg.northwestern.edu to listen to past episodes of the show and claim continuing medical education credit. And also be sure to subscribe to this show on Apple Podcasts or wherever you download your podcasts so that you never miss an episode. Thanks for listening.